Welcome to the Deep Dive, where we take your sources and really pull out the most important nuggets of knowledge.
That's the goal.
Today, we're diving deep into motor mechanisms and behavior from Campbell Biology in focus. Our mission is basically to unpack the how and why of animal activity. From the tiny dance of proteins in muscle cells, right up to big evolutionary dramas behind social behaviors.
Exactly. We'll explore how muscles and skeletons work together, how sensory inputs drive what animals do, how they learn, how they adapt, and how natural selection shapes pretty much everything, foraging, mating, even, well, altruism. You'll get a solid shortcut to understanding these key biological ideas and why they actually matter.
Right. So let's really unpack this. What are the fundamental building blocks of, well, all animal movement, and how do these little mechanisms scale up to create the amazing behaviors we see everywhere? You think about, say, a frigate bird soaring for days, or a tiny mouse using just its whiskers to navigate. It's incredible, and it all starts somewhere specific.
It really does. And that starting point, fundamentally, is muscle activity.
The engine, basically.
Precisely. And the core mechanism is amazingly almost universal across the animal kingdom. It relies on this intricate interaction between two main protein structures. You've got thin filaments, mostly made of a protein called actin, and thick filaments, which are these staggered arrays of myosin molecules.
Actin and myosin. Okay. So how are they arranged inside a muscle to actually, do something?
Good question. Imagine your muscle as a big cable. Inside are bundles of long muscle fibers. Each fiber is actually a single cell, just very long and with multiple nuclei. And inside each fiber, you find bundles called myofibrils. These are where the action happens.
Oh, okay.
These myofibrils are made of repeating sections called sarcomeres. Think of a sarcomere as the basic contractile unit. It's like a tiny engine. The thin actin filaments are anchored at the ends, the Z lines, and the thick myosin filaments are anchored in the middle, M line. They overlap. And this very precise arrangement, this overlapping pattern, is what gives skeletal muscle that striped or striated look you see under a microscope.
Okay, so the sarcomere is the engine. You mentioned they overlap. How does that lead to the whole muscle getting shorter? Do the filaments themselves shrink?
That's the fascinating part. They don't. This is the core of the sliding filament model. The actin and myosin filaments maintain their length. Instead, they slide past each other. The thin filaments are pulled towards the center of the sarcomere by the myosin filaments.
Like telescoping poles collapsing.
Exactly like that.
Yeah.
Or imagine interlocking your fingers and pulling them closer together. The filaments slide, the sarcomere shortens, and because all the sarcomeres are lined up, the whole muscle fiber shortens.
And the myosin is doing the pulling. How does that work? Are there little hooks?
Sort of. Each myosin molecule has a long tail and a globular head. This head region is key. It binds to ATP, our energy currency. When ATP breaks down to ADP and phosphate, the myosin head gets energized, kind of like cocking a spring. Then this energized head attaches to the actin filament, forming what we call a cross bridge. The release of ADP and phosphate triggers the power stroke. The myosin head pivots, pulling the actin filament along. It's like rowing a boat.
Pulling it towards the center.
Pulling it towards the center, yes. Then a new ATP molecule binds to the myosin head, causing it to detach from the actin. The cycle repeats: attach, pull, release, re-energize. This happens thousands of times per second across millions of myosin heads in a muscle fiber. That's what generates the force.
Wow. That's a lot of ATP being used constantly.
Mm.
How do muscles keep up with that demand? They can't store huge amounts, right?
Right. Storing lots of ATP directly is inefficient, so muscles have a layered system. For really quick bursts, like the first few seconds of intense activity, they use creatine phosphate to quickly generate ATP.
Like a quick energy shot.
Exactly. It lasts maybe 15 seconds. For longer efforts, they rely on breaking down stored glycogen into glucose and using aerobic respiration, which needs oxygen but can sustain contractions for, maybe up to an hour of moderate activity.
Okay, so we have the sliding filaments of power stroke, the energy supply. But how does the muscle know when to start this whole process? What's the switch?
Ah, the switch. It's a beautiful piece of biological engineering, and the key player is calcium. Calcium ions, K2 plus, B.
Calcium, like in milk.
The very same element, yes, but used here in a very specific way. At rest, when the muscle is relaxed, there are regulatory proteins on the actin filaments. Think of them as guards. Tropomyosin covers the sites where myosin wants to bind, and the troponin complex holds tropomyosin in place. So myosin can't bind.
Blocked.
Now the trigger comes from a motor neuron. It releases a chemical signal, acetylcholine, which causes an electrical impulse and action potential to spread across the muscle fiber membrane. This action potential travels down specialized tunnels called T-tubules deep into the fiber, right next to an internal storage network called the sarcoplasmic reticulum, or SR.
Okay, the SR, that sounds important.
It is. It's packed full of calcium ions. The action potential triggers the SR to open its calcium channels, flooding the cell's interior, the cytosol, with calcium.
So calcium floods out, and that's the signal.
That is the direct signal. The calcium ions bind to the troponin complex. This binding causes troponin to change shape, and when troponin changes shape, it pulls tropomyosin away from the myosin binding sites on actin.
Uncovering the binding sites.
Precisely. Now the myosin heads can grab onto actin, form those cross bridges, and start the power strokes. Contraction begins.
And relaxation.
When the nerve signal stops, calcium is actively pumped back into the SR. Its concentration in the cytosol drops sharply. Without calcium bound, troponin goes back to its original shape, allowing tropomyosin to cover the binding sites again. Myosin lets go, and the muscle relaxes.
It's incredibly elegant. And when this system goes wrong, that's linked to diseases.
Absolutely. Diseases like ALS, amyotrophic lateral sclerosis, damage the motor neuron so the signal never gets there, leading to muscle wasting. In myasthenia gravis, the body attacks the acetylcholine receptors, impairing the initial signal transmission. Understanding this calcium switch is crucial for understanding these conditions.
Wow. Okay, so that explains the on-off. But how do we control the force? I mean, lifting a feather versus lifting a heavy weight requires different amounts of effort.
Right. It's not all or nothing.
Yeah.
Our nervous system creates graded contractions. It adjusts the strength in two main ways. First, recruitment of motor units. A single motor neuron connects to multiple muscle fibers. That neuron and the fibers it controls are called a motor unit.
Got it.
To generate more force, your brain simply recruits more motor units. More fibers contracting means more overall force.
Makes sense. And the second way?
The rate of stimulation. A single action potential causes a brief, weak contraction called a twitch. If action potentials arrive faster before the muscle fully relaxes, the twitches start adding up. This is called summation. If the signals come really fast, you get a smooth, sustained, strong contraction called tetanus. Not the disease, just the physiological state.
Cessation and tetanus. Okay.
Yeah.
So we can control force by how many fibers are active and how fast they're being told to contract.
Exactly. It allows for incredibly fine control over muscle tension.
Now, you mentioned earlier that not all muscle fibers are the same, like dark meat versus white meat in a chicken.
That's a perfect example. Skeletal muscle fibers are classified based on how they make ATP and how fast they contract. You have oxidative fibers. They rely on aerobic respiration, so they have lots of mitochondria, a rich blood supply, and a protein called myoglobin that stores oxygen.
Making them look red, like dark meat.
Exactly. That myoglobin gives them the reddish color. They're built for endurance and resist fatigue. Think marathon runner muscles. Then you have glycolytic fibers. They rely more on glycolysis for quick ATP production. They tend to have a larger diameter, less myoglobin, hence white muscle, and they fatigue much faster. Think sprinter muscles.
And fast twitch versus slow twitch.
That refers to contraction speed. Fast twitch fibers develop tension more rapidly. Slow twitch fibers contract more slowly, partly because they pump calcium back into the SR more slowly, so the twitch lasts longer. Generally, all slow twitch fibers are oxidative. Fast twitch fibers can be either oxidative or glycolytic, offering different combinations of speed and endurance.
So different muscles in the body will have different proportions of these fiber types depending on their job.
Precisely. Some animals even have incredibly specialized fibers, like the super fast muscles rattlesnakes use for rattling, or certain fish use for mating calls hundreds of contractions per second.
Amazing. And beyond skeletal muscle, there's cardiac and smooth muscle.
Right. Cardiac muscle, found only in the heart, is also striated, but the cells are branched and connected by intercalated discs. These discs allow electrical signals to pass directly between cells. This means the heart can initiate its own rhythmic contractions, and it prevents the kind of tetanus you see in skeletal muscle, which would obviously be fatal in the heart.
Obviously. And smooth muscle.
Smooth muscle is found in the walls of hollow organs, think your digestive tract, blood vessels. It lacks striations, contracts, and relaxes much more slowly than skeletal muscle, and uses a different protein, calmodulin, to handle the calcium signal instead of troponin. It's adapted for sustained, involuntary contractions.
Okay, that's a fantastic overview of the muscle engine itself. But muscles don't work in isolation, right? They need something to pull against.
Exactly. That's where skeletons come in. They are the essential partner to muscles. Skeletons provide the rigid framework for muscle attachment, allowing movement. They also support the body against gravity and protect delicate internal organs like your skull protecting your brain.
And usually muscles work in pairs, don't they?
Yes, typically as antagonistic pairs. One muscle contracts to cause a movement while its partner relaxes. To reverse the movement, the roles switch. Think of your biceps bending your elbow and your triceps straightening it.
Simple but effective. And you
Yeah.
mentioned different types of skeletons earlier, not just bones like ours.
Correct. There are three main types. First, hydrostatic skeletons. These are found in soft-bodied animals like earthworms or jellyfish. It's basically a compartment filled with fluid under pressure. Muscles contract against this incompressible fluid. Earthworms use alternating contractions of circular and longitudinal muscles along their segments to move. That's peristalsis. Even the human penis uses hydrostatic principles for erection.
Interesting. Okay, what's next?
Exoskeletons. These are hard external coverings. Think of a clam shell made of calcium carbonate or an insect's cuticle made of chitin and protein. Exoskeletons provide great protection and leverage points for muscles, but the animal has to shed it molt to grow bigger, which is a vulnerable time.
And finally, the ones we have.
Endoskeletons. Hard supporting structures inside the body buried within soft tissues. Examples range from the simple spicules and sponges to the bony skeletons of vertebrates like us. Our skeleton has over 200 bones connected by ligaments at joints like ball and socket joints in the shoulder, hinge joints in the knee, pivot joints in the neck, allowing for a huge range of motion.
So muscles provide the power, skeletons provide the structure and leverage. But putting it all together for locomotion moving from A to B, that seems like a real engineering challenge.
It absolutely is. Locomotion requires expending energy primarily to overcome two forces, friction and gravity. The relative importance depends on the environment.
Okay, let's take land first.
On land, gravity is a major challenge. You need strong skeletal support and powerful muscles. Balance is also key. Kangaroos are a fantastic example. Their huge hind leg muscles and elastic tendons store and release energy with each hop, making it incredibly efficient. Their tail acts like a third leg for balance when standing still.
And crawling, like snakes or worms.
To crawlers, friction is often the main hurdle. Earthworms use that peristalsis we mentioned. Snakes use various methods like undulating their bodies side to side or using belly scales to grip and push.
What about in water? Swimming seems different.
In water, buoyancy largely counteracts gravity, which is a huge energy saver. But drag, or friction from the water, becomes the biggest problem. That's why you see so many aquatic animals with a sleek, fusiform, or torpedo-like body shape, think tuna or dolphins. It minimizes drag. They might use limbs as oars or undulate their bodies or tails to propel themselves.
And flying. That seems the hardest.
Flying requires overcoming gravity by generating lift and also minimizing drag. Wings act as airfoils, shaped to create pressure differences that generate lift. Birds have amazing adaptations for flight. Hollow bones, no urinary bladder, no teeth, anything to reduce weight. Their streamlined shape minimizes drag too.
Is there a most efficient way to move?
Interestingly, yes, generally speaking. Based on energy cost per unit of body mass per distance traveled, swimming is usually the most efficient.
Really? Because of buoyancy?
Largely, yes. Flying is next, and running or walking on land is typically the least energy efficient, mainly due to constantly fighting gravity. Also, larger animals tend to be more efficient movers than smaller ones within each mode of transport.
Fascinating stuff. Okay, so we've covered the mechanics, the muscles, the skeletons, the physics of movement. Now let's shift gears a bit. How does this physical capability translate into behavior? The things animals actually do.
Right. This moves us into the realm of ethology, the study of animal behavior. And a great framework comes from Nico Tinbergen, one of the pioneers. He proposed 4 key questions to understand any behavior.
4 questions. What are they?
They split into 2 categories. Proximate causation asks how a behavior occurs, what stimulus triggers it, what physiological mechanisms, nerves, muscles, hormones are involved. Ultimate causation asks why a behavior occurs in terms of evolution. How does it help the animal survive and reproduce? What's its evolutionary history?
How versus why.
Right.
Okay, let's start with some hows. Sometimes behavior seems almost automatic.
Exactly. Those are often fixed action patterns or FAPs. These are sequences of innate, unlearned behaviors that are essentially unchangeable. Once triggered by a specific external cue called a sign stimulus, they usually run to completion.
Like flipping a switch.
Pretty much. The classic example is the male three-spined stickleback fish. During mating season, they attack anything with a red underside. Researchers found they'd attack crude models fiercely if they had red, but ignore realistic fish models without red. The red color itself is the sign stimulus triggering the aggressive FAP.
So it was a hardwired response.
Yeah.
What about more complex behaviors like migration, animals traveling huge distances?
Migration is this regular long-distance change in location. It's incredible. And animals navigate using a variety of cues.
Like what? How do they know where to go?
Many use the sun's position, but they have to compensate for its movement across the sky using an internal circadian clock, their daily biological rhythm. Others navigate by the stars, and some, remarkably, can detect and orient to the Earth's magnetic field. Pigeons, sea turtles, even some bacteria use this magnetic sense.
A built-in compass. And you mentioned circadian clocks. Timing is important too, right?
Rhythms. Absolutely. Behavior isn't just about where, but when. Behavioral rhythms are critical. The circadian rhythm governs daily cycles of sleep, activity, feeding, hormone release, etc. It's synchronized by light and dark, but it persists even in constant conditions.
And longer cycles.
Yes, circannual rhythms are linked to the yearly cycle of seasons, influencing things like migration and reproduction timing. These are often cued by changes in day length. And some behaviors are even linked to lunar cycles, like the courtship of fiddler crabs peaking around new and full moons when tides are strongest, which helps disperse their larvae.
Wow. Okay, so animals navigate, they have internal clocks. How do they interact with each other? Communication must be key.
Hugely important. We define a signal as a stimulus transmitted from one organism to another, and communication as the transmission and reception of those signals.
How does that work? Is it just sounds?
Oh, it's incredibly diverse. Think about fruit fly courtship. It's a whole sequence, a stimulus-response chain. First, the male sees the female. Then he uses his sense of smell olfaction to detect species-specific chemical signals, pheromones. Then he taps her with his leg tactile. Finally, he vibrates a wing to produce a courtship song auditory.
A whole conversation.
Exactly. And mating only happens if all the signals in the chain are performed correctly and received appropriately. The form of communication often matches the animal's lifestyle and environment. Nocturnal mammals might rely heavily on smell and sound. Diurnal birds often use bright visual signals and complex songs.
Pheromones can be really powerful, right?
Incredibly so. A female silkworm moth releases pheromones detectable by males kilometers away.
And maybe the most famous example of complex communication, the honeybee dance.
Ah, yes. Carl von Frisch's amazing discovery. Foraging honeybees perform a dance language back at the hive to tell other bees about food sources. A round dance means food is nearby, within about 50 meters. No direction info needed. But for distant food, they perform a waggle dance. It's a figure-8 pattern. The duration or number of waggles in the straight run part indicates the distance.
And a direction.
The angle of that straight run relative to the vertical surface of the honeycomb indicates the direction of the food source relative to the current position of the sun outside the hive. It's truly symbolic communication.
Absolutely incredible. Okay, so we have innate behaviors, complex communication, but animals also learn, right? It's not all pre-programmed.
Definitely not. This brings us to the classic nature versus nurture debate, but really it's almost always both. Innate behavior is developmentally fixed, but learning is the modification of behavior based on specific experiences. Learning involves actual changes in the nervous system, the formation of memories through altered connections between neurons. The key is understanding how genes and environment interact to shape behavior.
How can scientists tease apart those influences?
One powerful technique is the cross-fostering study. You take the young of one species and have them raised by adults of another species. For example, researchers studied 2 species of mice. California mice males are aggressive and very paternal. White-footed mice males are less aggressive and offer little parental care.
Okay.
When California mice pups were raised by white-footed mouse parents, they grew up to be less aggressive and less attentive to their own pups compared to those raised by their own species. It shows a strong environmental influence on these behaviors.
So early life experience really matters. What about other forms of learning? There's imprinting, isn't there?
Yes, imprinting is fascinating. It's a type of learning that forms a long-lasting behavioral response to a specific individual or object, but it only happens during a limited sensitive period, usually early in life. The classic example is Konrad Lorenz and his geese. Goslings imprint on the first moving object they see shortly after hatching, usually their mother. But Lorenz showed they'd imprint on him and follow him around if he was the first thing they saw.
Does that have practical uses?
It does, especially in conservation. For instance, captive-bred whooping cranes need to be raised carefully. If they imprint on humans, they won't mate with other cranes. So handlers might wear crane suits or use puppets to ensure proper imprinting, helping the birds integrate into the wild.
Clever. What about learning about the environment itself, finding your way around?
That involves spatial learning, establishing a memory of the environment's spatial structure, locations of nests, food, water, potential mates, hazards. Tinbergen did another classic experiment with digger wasps. They nest in hidden burrows. He placed a ring of pine cones around a nest entrance while the wasp was inside. When she emerged, she flew off.
And then?
While she was gone, he moved the ring of pine cones a short distance away. When the wasp returned, she flew to the center of the pine cone ring, not to her actual nest entrance. It showed she'd learned the arrangement of landmarks.
So she made a mental map.
Essentially, yes. More complex spatial learning involves forming a cognitive map, an internal representation or code of the spatial relationships between objects in the environment. Clark's nutcrackers, birds that cache thousands of seeds, seem to use cognitive maps, sometimes finding caches located halfway between specific landmarks.
Like having a built-in GPS. What about linking two different things together, like cause and effect?
That's associative learning, the ability to associate one environmental feature with another. For example, a blue jay eats a monarch butterfly, which tastes foul and makes it vomit. The jay quickly learns to associate the bright orange and black pattern, feature 1, with the unpleasant experience, feature 2, and avoids monarchs in the future.
But can animals associate anything?
Interestingly, no. There seem to be evolutionary constraints. Pigeons can easily learn to associate danger with the sound, but not with the color. They associate food with color, but not sound. Rats quickly associate illness with unfamiliar smells, but not with sights or sounds. It makes evolutionary sense these predispositions reflect the likely cause and effect relationships in their natural environment. A bad smell is more likely linked to bad food than a flashlight is.
Right, the learning is biased towards what's ecologically relevant. And then there's the really complex stuff, thinking, problem solving.
Yes, cognition in its broadest sense includes awareness, reasoning, recollection, judgment. It was once thought to be uniquely human, or maybe limited to primates, but we now see cognitive abilities in many animals, even insects like honeybees who can learn concepts like same versus different.
And problem solving.
That's the process of devising a strategy to overcome an obstacle to reach a goal. Think of a chimpanzee stacking boxes to reach a banana hanging from the ceiling, or a raven figuring out how to pull up a string piece by piece to get food tied to the end.
And they can learn by watching others do it.
Absolutely. That's social learning. Young chimpanzees learning how to crack nuts by watching experienced adults is a prime example. This transmission of learned behaviors through observation or teaching is the basis of culture in animal populations' information transfer that influences behavior across generations.
This is amazing. It really bridges the gap between the mechanics and the why, which brings us back to evolution. How do specific behaviors help an animal survive and reproduce?
Great question. Let's look at foraging behavior, everything an animal does to find, recognize, and capture food. Natural selection should favor foraging strategies that maximize energy gain while minimizing energy cost and risk.
Can we see this evolving?
Yes. A fantastic example comes from fruit fly larvae and a gene called forager. There are two main alleles. 4R, rover, makes larvae travel farther while feeding, while 4S, sitter, makes them move less. In lab populations kept at low density, the forest allele became more common. Why waste energy moving if food is plentiful? But in high-density populations, the 4 allele increased. It pays to explore when resources are scarce nearby. This shows direct density-dependent selection acting on a behavioral trait.
Oh, evolution in a lab dish. Okay, foraging is crucial for survival. But for passing on genes, mating behavior is key.
Absolutely. Mating behavior and mate choice encompass attracting mates, choosing among potential partners, competing for them, and sometimes caring for offspring. These behaviors are under intense selective pressure.
And this leads to different mating systems.
Exactly. Systems range from promiscuous, with no strong pair bonds, to monogamous, one male and one female pairing off, often looking similar, to polygamous, where one individual mates with several others. Polygamy can be polygyny, one male, many females, males often larger or showier like elk, or polyandry, one female, many males, females can be the showier sex like in some shorebirds.
What determines which system evolves?
A major factor is the needs of the young. If offspring require a lot of care, perhaps continuous feeding from both parents, monogamy might be favored. If young are relatively self-sufficient soon after birth or hatching, one parent, often the male, might maximize their reproductive success by seeking additional mitts, leading to polygyny.
And parental care itself, who does it?
That also varies hugely. In mammals, females often provide the bulk of care due to lactation. In birds, where chicks often need feeding by both parents, monogamy with shared care is common. A fascinating factor influencing male care is certainty of paternity.
How sure the male is that the offspring are actually his.
Precisely. With internal fertilization, like in mammals and birds, mating and birth hatching are separated in time, so paternity certainty can be relatively low for the male. This might explain why exclusive male care is rare in these groups. Males might instead guard their mates or compete with other males. But with external fertilization common in many fish and amphibians, mating and egg-laying often happen almost simultaneously. The male knows he fertilized those eggs. Here, paternity certainty is high, and male parental care is just as common, if not more so, than female care. Think of a male fish guarding a nest of eggs.
It's not about conscious thought, but about selection-favoring behaviors that maximize the spread of one's genes.
Exactly, and this ties into sexual selection, which drives the evolution of traits related to mating success, often leading to sexual dimorphism, those differences in appearance between males and females.
Like bright colors or large antlers.
Right. Intersexual selection is mate choice. Usually females choosing males based on certain traits, courtship songs, flashy colors, elaborate displays that might indicate male health or quality. Intersexual selection is competition within one sex, usually males fighting or displaying for access to mates. Think of rams battling, or those stalk-eyed flies comparing eye stalk length.
Okay, so evolution shapes foraging, mating. Let's drill down into the genes again. How specifically can genes influence complex behaviors, and how does evolution explain apparent selflessness or altruism?
We're learning more and more about the genetic basis of behavior. Sometimes surprisingly complex behavioral differences can be traced back to variations in a single gene.
Really? Like what?
The vole example is classic. Prairie voles are monogamous, form strong care bonds, and males are very paternal. Meadow voles are solitary and promiscuous with little male care. The difference seems largely due to a brain chemical, the neurotransmitter vasopressin, and specifically the distribution of its receptors in the brain. Prairie voles have many more vasopressin receptors in key brain areas associated with reward and bonding.
And they tested this.
They did. Researchers experimentally increased vasopressin receptor levels in the brains of meadow voles, and remarkably, these voles started showing behaviors much more like prairie voles' increased pair bonding and paternal care. It's strong evidence for a direct genetic link to complex social behavior.
That's incredible. And we can see genes and environment interacting in wild populations too.
Yes. When you find behavioral variation within a species that correlates strongly with different environmental conditions, it suggests past natural selection has shaped those behaviors. Remember the garter snakes. Coastal snakes eat slugs, inland snakes don't. Lab studies showed this preference is innate. Young snakes raised in the lab showed the same preference as their wild parents from either location.
So it's genetic.
It appears to be a genetically acquired taste, likely linked to the ability to detect certain chemicals from the slugs. The interpretation is that when garter snakes originally colonized coastal areas from inland, individuals who happened to have mutations allowing them to recognize and eat the abundant slugs had a survival advantage. Over time, selection favored this trait, leading to the distinct dietary preference in coastal populations.
Evolution in action. Okay, final big puzzle. Altruism. Why would an animal do something that lowers its own chances of survival or reproduction but helps another? Like a squirrel giving an alarm call that draws attention to itself.
It seems paradoxical from a simple survival of the fittest viewpoint. Altruism is defined as a behavior that reduces the altruist's individual fitness but increases the fitness of other individuals in the population. Naked mole rats are another example. Non-reproductive individuals defend the colony, even sacrificing themselves.
So how does evolution allow for that?
The key insight came from William Hamilton and his concept of inclusive fitness. An individual's genetic success isn't just measured by its own offspring. It also includes the reproductive success of its close relatives, because they share many of the same genes.
So helping your relatives helps your own genes pass on indirectly.
Exactly. Inclusive fitness is the total effect an individual has on proliferating its genes, both by producing its own offspring and by providing aid that enables close relatives to produce offspring they might not otherwise have had. This led to Hamilton's rule, a mathematical prediction for when altruism should be favored by natural selection. RBC.
Okay, break that down. REC.
R is the coefficient of relatedness, the fraction of genes shared between the altruist and the recipient. R equals 0.5 for siblings or parent offspring. R equals 0.25 for half-siblings or uncle-niece. R 0.125 for first cousins. B is the benefit to the recipient, the average number of extra offspring the recipient produces because of the altruistic act. C is the cost to the altruist, how many fewer offspring the altruist produces as a result of performing the act.
So, if the benefit to the relative, weighted by how related they are, outweighs the cost to yourself.
Then natural selection favors the altruistic behavior. This specific type of natural selection is called kin selection. It explains why altruism is often directed towards close relatives. The squirrel giving an alarm call might endanger itself slightly, C is small, but if it saves several siblings or offspring, RU 0.5, B is large, the condition RBC is likely met.
As Haldane famously joked, he'd lay down his life for 2 brothers or 8 cousins.
Precisely captures the logic of kin selection. It weakens rapidly with decreasing relatedness.
Okay, so pulling this all together, we've gone from the microscopic actin and myosin sliding past each other right up to the evolutionary logic behind complex social strategies like altruism. It's an incredible journey.
It really is. Every animal action, from the simplest twitch to the most complex migration or courtship display, is underpinned by these biological mechanisms shaped over evolutionary time.
So the key takeaways for everyone listening.
I'd say. First, animal movement relies on the fundamental actin-myosin interaction, regulated by calcium and the nervous system. Second, skeletons hydrostatic, exo, or endo provide the crucial framework for locomotion adapted to specific environments. Third, behavior is this dynamic mix of innate programming and learned experiences driven by sensory input, communication, and biological rhythms. And finally, evolution through natural selection, sexual selection, and kin selection, explained by inclusive fitness, is the ultimate architect, shaping why animals forage, mate, and interact the way they do.
Which leaves us with a final thought to chew on. Given these deep biological and evolutionary roots of behavior, both genetic and environmental, how much of your everyday life, how you learn, how you make decisions, how you interact with family and friends might be influenced by these same fundamental forces? What connections can you maybe spot now that you couldn't before?
It's definitely food for thought. Thank you for joining us on this deep dive. We hope you feel better informed, maybe even a bit inspired to look at the living world, including ourselves, with a fresh perspective.
Absolutely. Until next time, keep exploring, keep questioning, and keep diving deep.